Hello everyone, and my name is Danielle. My project was on the effects of herbivories on spekboom rooting and leaf moisture content. It is important to understand the effect herbivory has on such a plant so that the result can be applied to knowledge of overgrazing and how different types of herbivories can affect the shrub. Overgrazing is estimated to have degraded 46% of the naturally occurring spekboom thicket alone. Spekboom is an iconic plant in South Africa which is also a common food item for many native herbivores. It has also been used in degraded thickets for restoration or to improve the soil properties and the vegetation density. Our experiment was conducted on the Nelson Mandela University South Campus to determine how different types of herbivories affect this shrub. The effects were measured by leaf moisture content and a root rating based on the length and number of roots present. The hypothesis is that plants that suffer more damage under herbivory, which was the 50% biomass loss treatment, would have a lower leaf moisture content and root development when compared to the control, as the plant will have to spend more resources on recovering from this significant herbivory rather than on new growth. Spekboom fingerlings were harvested from the botany garden and planted into 2 seedling trays. These seedling trays were placed on the ground outdoors. After 10 days, the trays were experimented with by altering the fingerlings in ways that could result in delayed or accelerated rooting and affect leaf moisture content. These treatments were based on and meant to simulate herbivory. We did have a control treatment. The other 3 treatments included 50% foliage removal, a 50% biomass removal, and the removing of the tip of the fingerlings. On day 49, after the 2 trays underwent their treatments, they were moved into the growth chambers inside of the science building. The machines allow for researchers to program a climate specific for their study purposes. Temperature, light intensity, and wind were all set on a timer. After 24 days in the growth chamber, the fingerlings were removed and then gently removed from the trays. The roots of each fingerling were photographed and rated on a scale from 1 to 5. A rating of 1 indicated little to no roots were present, and a rating of 5 would indicate numerous large roots were present. The leaves of each fingerling was removed and placed into a numbered beaker. The beakers were numbered in order to keep track of the root rating for each fingerling as well as the wet and dry mass of the leaves. The wet weight of the leaves were measured on the scale. All the beakers were then placed into an oven for 3 days, and after drying, the leaves were weighed again to obtain the dry mass. For the mass of the leaves before and after drying, you can see by these graphs that the results followed a similar trend, where the tip removal had a highest mass before drying, it would have the highest mass after drying, and that same trend would follow for most of the things across the treatments and across, the maximum, minimum, the average, and also the mode. But looking at specifically the leaf moisture content, which is one of the things that we're focusing on here, you can see that there wasn't a significant difference between any of the treatments. And a NOVA test was also run, and it showed that there was no significant difference between the different types of herbivories, with a p-value of above 0.05. The degrees of freedom also indicated that there was no difference between the means of the different herbivories. As was the case with the leaf moisture content, the root rating showed no significant difference between the treatments, and a chi-square test failed to reject the null hypothesis. So in conclusion, herbivories did not have any significant effect on the leaf moisture content and the root rating of spekboom.